This is the CBE 205 final project presentation for Alec Brickell, Saad Al-Harbi, and Blessing Atanga on mechanobiology. Mechanobiology is a fairly new field which caught the eye of the scientific community after Darcy Thompson's publication on growth and form in 1917. Within his book, he described his idea that mechanical forces can influence the shapes of plants and animals. Mechanobiology is an interdisciplinary field combining biology, physics, and mechanics to understand cellular structures and physiology. One such example is muscle building. Muscle tissue is able to sense the tension placed upon it and respond with biological pathways to increase the tissue and to be able to better withstand further mechanical activity. Mechanobiology is the study of how mechanical forces, including both stretching and squeezing, influence the cells at a cellular, molecular, and tissue level. This helps us understand how the cells will interact with their surroundings, sensing and responding to factors, including even the stiffness of their environment. At a cellular level, mechanobiology is about how cells deal with and respond to physical forces. When we look closer at the molecules, mechanobiology is essential because it explains how certain molecules come together and work to activate specific functions in the cell. These processes happen because of two main things. The first being mechanosensing, which means the way that cells are able to sense the physical signals and forces from their surroundings, which in turn activates mechanotransduction, where cells are able to turn these external forces into biochemical signals to make certain cell functions happen. Mechanobiology is very important for understanding disease. Abnormal mechanobiological processes can contribute to the development and progression of various diseases, such as cancer and cardiovascular disease. But since mechanobiology reveals how cells respond to physical forces and cues, studying mechanobiology provides insight into disease mechanisms and opens avenues for developing targeted therapeutic interventions aimed at modulating mechanical aspects to treat or prevent disease. There are various techniques which can be used to measure the mechanical properties of single cells, including their elasticity and viscosity, which serve as key indicators of a cell's mechanical characteristics. These techniques include things such as atomic force microscopy, micropipette aspiration, optimal tweezer, and magnetic twisting cytometry. The mechanobiological process begins with focal adhesion molecules, which dictate mechanical stimuli to integrins in the transmembrane region. This activates cellular pathways such as the Roa-Rock pathway, which combine with activation of ion channels to relay the perceived information. The pathways lead to changes in the nucleus represented by chromatin remodeling and changes in the lamin of the cell nucleus. When the pathways reach the nucleus, they predominantly affect the nuclear lamin and chromatin remodeling. The DNA will alter the way in which it is wrapped around histones, changing between the more densely packed heterochromatin and the more genetically available euchromatin. As a result, the mechanosensitive pathways alter the genetic expressions of cells. Several tools can be used to quantify these changes, and in turn better understand the effects of the mechanobiological pathways. One such tool is the application of displacement microscopy to calculate a strain map based on a displacement field of the cell's nucleus. This helps us to understand how the nuclear chromatin is changing over time. These pathways are actively being researched, both with the intent of increasing understanding, as well as developing therapeutics that will help solve medical problems. The image on the left helps us to understand the consequences of laminar degeneration, as we see that the mice cells lack structure, and the mouse presents with a smaller phenotype. This is evidence that laminar degeneration accelerates aging. The image on the right displays the difference between H3K9ME3, an epigenetic histone modification marker, in contractile and non-contractile cells. It is clear that the contractile cells, which are subject to mechanical forces, are able to more effectively maintain their important epigenetic chromatin remodeling pathways and are additionally more resistant to fibrosis. This research helps us understand and develop treatment for pathologies such as laminopathy, as well as understanding the importance of mechanical stimuli on overall cell health. One final example is the importance of mechanobiology is the correlation between substrate stiffness and mesenchymal stem cell differentiation. We see that on a soft substrate, the cells will become neurons. On stiff substrates, the cells will become osteocytes. And on medium substrate, they will differentiate into myocytes. This helps us to understand growth and regeneration pathways and avoid medical disasters, such as stem cell differentiation into osteocytes when implanted in hearts, by first subjecting the therapeutic cells to their proper mechanical environment. Thank you for listening, and we hope that we effectively communicated how mechanobiology is an exciting, emerging interdisciplinary field, which presents itself to interesting research and important therapeutic applications.